it is connected to this topic. Is Anastasia here?
Hello everyone.
Hi, Anastasia.
I'm really happy to see you despite online format. Thank you for the organization and for inviting me to participate. So I'm ready for the question.
Can you share your poster?
Yes, I will try.
Sorry, Zoltan, you wanted to ask something?
No.
No.
Yes, I
sorry.
wanted to add a comment, but another time. I don't want to steal your time.
No, it's not a problem. I'm sorry. The moderator, I can't see. I didn't see your reaction. I'm sorry that I'm not on time.
Right, we can talk about this. David's material. I wanted to mention another interesting phenomenon, but we can talk about this another time.
Can you please just put it in the chat, because I want other people, of course, to see your comments, and we can chat on this later. I'm sorry. So Anastasia, I wanted, because we were speaking about oxidative stress and changes in the organism, and there are theories about cancer, that it is basically cancer and diabetes, that these are the just related to stress. So you did work about cancer detection, and that is a unique poster in this conference, that it is related to medical diagnostics. So can you tell us something interesting about your results regarding different body fluids?
Yes, I can tell a little bit. I'm just on at the beginning of my aquaphotomics journey. So I try to understand more in aquaphotomics. Thank you, Jelena, for guiding me. So what we have 3 groups of fluids, plasma, urine, and serum. And as we can see on the spectra, the main first overtone, there is the data of scale. That's the reason we analyze 4 intervals of the 4 intervals, and I built the model for each intervals for each patient. So what we can see on subtracted spectra, that all the fluids have almost the same wavelengths. So it could be coherence together. But after PCA analysis, I got a lot of data, a lot of loadings and peaks. And here we can see the most most peaks which I found in subtracted spectra and in PCA loadings. There is more than I showed here, but it is too much to put it on this table. So I found some wavelengths peaks for just for plasma, just for urine, but many peaks which the same or close together for all fluids. But when I tried to build aquagram for all data, it showed big baseline difference. So we suppose that maybe better to build aquagram for each region, for each patient, and try to find difference in cancer and in data before surgery and after surgery.
Nastya, could you please make your poster a little bit smaller so we can see it, the whole one?
Yes.
Oh, okay. Okay, thank you. It is very interesting because years ago we found in inflammation of mammary gland the same effect in blood and urine and milk. So it's kind of normal, but it's very good news. So when you do subtraction, do you subtract what do you subtract?
I subtract all average spectra before spectra of the samples before operation and all the samples after operation.
Okay.
Surgery.
Okay, understand. Yes, now I remember. Thank you.
Mm-hmm.
Happy to see you again.
Happy to see you too.
Yes, I'm sorry that you couldn't come. It was really unlucky that we didn't have time to organize, as you were so brave and wanted to come.
Oh, thanks, Jelena.
No, this time is very difficult time.
I hope I will have reason to come again.
Yes, I also hope. And what I like to point out about your poster, I used your part of your spectra in the workshop to show that people. So I really wanted to show that everything can be used. Just look for the parts. Nastia, we have a question from Pierre to you. Mm-hmm. Do you see? Do you see in the chat? So please just show. So Pierre asks, dear Anastasia, was this just surgery, no chemo or radiotherapy applied?
Yeah, it was surgery, but I'm not sure about chemotherapy because the data was applied by our colleagues in Germany. And unfortunately, we haven't all details about the data. So, and there are different cancers on this data. For example, malignant mesothelioma, appendix cancer, sigma carcinoma, and others. So it's different type of cancer. Maybe that's the reason we can see on the spectra two groups, but groups not for only for before and for after surgery. It's different because of patients.
Yeah, patients. Nisha was talking something about the patients. Professor Surkova touched a little bit about this individuality. Pierre, why do you ask this question? Pierre, are you here? Can you?
Oh, hello.
Ah, thank you.
Hi.
I have the kids in the background, and it's pretty noisy, so that's why.
Oh, well, kids can join.
Okay. I'll write the question to Anastasia because there is some other issues that I may suggest this could be helpful in terms of, or maybe since the kids are quiet, let me see. Anastasia, is it possible to split up this group of samples into, let's say, one group has undergone chemotherapy, another one radiotherapy, and the third one just surgery, to make it a little bit more pinpointing to what was the effect of these three different therapeutic approaches?
It would be nice, but unfortunately we haven't this information.
Well, I think it can be done in the future, right?
Yes, it can be. Thank you for the advice.
Now, then there's the second one, because if that what you shows relates to the changes in the PCI structuring that we plotted up, the principal component analysis shows quite distinct differences. I wonder whether it has something to do with the decoherence of tumor cells. Well, tumor cells is an inappropriate term, but allow me to use it. Of these cells that go into decoherence, and then these cells detune the water clusters. And it all pins down to quantum electrodynamics.
Mm-hmm.
So it has a huge implication in terms of what you now present us and how we're going to use this information.
Oh, yes.
Yes, absolutely. Very good point. Thank you, Pierre.
Okay, I can switch off because the kids are in the background.
Yes, I understand, Pierre.
Thank you, Anastasia. Thank you.
Thank you too.
I'd also like to comment. Yes, please, Felix.
Just a quick point. I think these studies with cancer are very interesting, but the study design is, of course, critical. So I would start it in a different way. I would just at the first stage compare healthy populations with cancer patients. So collecting, for example, their urine and different bloods, I mean blood, serum and plasma from healthy subjects and cancer patients, and then create the aquaphotomics analysis. And then in the later stages, you can increase the analysis, because I think the study at the moment is a little bit difficult to interpret, because the surgery is causing, of course, a lot of stress on the organism, and with inflammation happening and so on, and these not necessarily are correlated to the cancer per se. So,
yes,
you're right.
We were aware of that, Felix, and that is exactly what I wanted to comment. The point is, with medical studies, and this is what I really hate, we are so limited by the information, by the samples that we can obtain. So, for example, I also could, I had serum and urine samples of the patients who underwent colorectal cancer surgery, and I didn't have healthy at all. I could have samples from those people who had colorectal, really confirmed cancer, and those who turned out to be benign, so just tumors. I did not have a proper control group, so it's very, very difficult to obtain. And regarding the anesthesia, you should have people, persons under anesthesia who are healthy, and then with the condition, until it's really difficult to work with human, unfortunately.
it is, yeah, but worth the effort, I would say.
Worth the effort. Yes, I really would love to include more. You work in the hospital, right? You can get us samples.
Felix, you've got a job now.
Another job, yeah.
Yeah.
Yes, I would love that. Anastasia, sorry, you have a question from Professor Atanasova. So she asks whether you have the spectra of urine, plasma, and serum for the same patients.
Yes, we have for the same patient. Just for 2 patients, we have data only for plasma. For urine, just 2 patients, we have just 1 fluid, and after others the same.
Yes, it's interesting to always have all fluids. I also wanted, when we did the mastitis study, I also wanted saliva, because it's just easier to lick. For example, even for detection of corona, we wanted to study that.
But, I was thinking about Felix's question. One thing that when we work with individual living systems, it is always better to start with showing the differences between systems. So I'm sure that the data that you have collected from different patients, if you do PCA or SIMCA or whatever, you can show a big difference from patient to patient. And then when you show a common, a consistent subtractive spectra, then we can start thinking about, okay, why they are similar. Is it oxidative stress, as Felix pointed out, that it might be stress? We don't know. Or then once we have data about the healthy patient and cancer patient, then we can see where this is closer to the healthy patients or to cancer patients. But the first step is always should be to show the differences, because once you have that big difference from patient to patient, and then you go and get similar subtractive spectra, then it obviously you have a perturber, a common influence that brings all these different systems to one similarity.
Thank you, Professor Surkova. I also did subtracted for each patient, and I just saw here for all data set, because there are a lot of graphs. So, but it looks similar, many bands in the subtracted spectrum.
Thank you, Nastya. Felix, did you want to ask or to comment something? I think
Oh, it's okay.
It's okay. All right, thank you. Any more questions, maybe from Nastya?
Jelena, I was wondering if we can continue tomorrow, for example.
I think we could. if our we have actually Felix Scholkman, Janet Boder, and John Louis Ziniazaku. Can they
today?
also attend? Yeah.
Okay.
Can they attend? Can you maybe confirm Janet and Felix and
okay.
John Louis? Can you confirm to me that you can attend tomorrow poster session or not?
I can.
You can.
Yeah.